I'm very excited to share a story of HIV research in my laboratory. And I think the idea is, how can we stop HIV and control viral infection in our cell, immune cell? 35, 36, 37. Those might be your lucky numbers. However, when we talk about HIV or AIDS, this is very sad numbers. 35 million people have died because of AIDS or related diseases. 36 years ago, HIV was first reported in the US in 1981. 37 million people currently living with HIV infection. HIV remains a global epidemic as of today. Just last year, there were more than 2 million people newly infected by HIV, and 1.1 million people died because of AIDS or related diseases. If you do a simple math, it's about 2 people died of AIDS per minute just the last year. However, the great news is the AIDS-related deaths have declined every year because increased HIV treatment coverage every year going up. In fact, during the last 36 years research, we had a major accomplishment. Scientists and physicians have transformed this devastating disease from death sentence into a fully manageable chronic disease. This is mainly due to very powerful anti-HIV therapy. However, we still face a lot of challenges as of today. First, we don't have a cure for AIDS. We don't have a very effective vaccine to prevent from infection. Second, although we have more than 30 FDA-approved anti-HIV drugs, all of those drugs can have drug-resistant problem because HIV gene mutation. Additionally, those patients need to take pills every day in their remaining life if we don't find a cure. Of course, those drugs can have side effects. 8 years ago, an HIV patient visited my lab because he was very interested in my research. He told me, over 12 years ago, he set up 3 timers every day to remind himself to take more than 14 pills every day to control his HIV infection. Nowadays, he and many HIV patients only take 1 pill a day. But inside of this 1 pill, there are more than 3 drugs, normally 3. You can imagine if they take this drug in their remaining lifetime, it can be toxic. I remember the patient told me how terrible the experience of feeling he had when he took more than 14 pills per day. However, HIV, the major problem is viral persistence. HIV is very good at hiding our immune cells, therefore avoiding immune control of the disease. When we think about HIV, it can be very complicated. However, HIV is ridiculously simple, yet astonishingly complex. Here I quote a sentence from a journalist of Science magazine, John Coyne. I think this word really covers the major feature of HIV. So how simple HIV can be. If we compare between HIV and a human cell, you can see HIV only has 9 genes, encoding 15 proteins. The viral RNA genome has only 9,700 bases. In contrast, our human cell has about 20,000 genes, which encode 18,000 proteins. Our genome DNA contains about 3.3 billion base pairs. We can use electron microscope to look at HIV. You can see this tiny particle, the diameter is about 100 nanometer or 0.1 micron, which is about 1,000th of the diameter of our hair. In contrast, a human cell, like we show here, a paper we published 10 years ago, we can see those immune cells, their diameter is about 25 micron, which is about 250 times bigger than the HIV particle. In fact, there are numerous HIV particles at the cell-cell junction in this photograph, but they are just too tiny to see. HIV can be very complicated when we think about a virus interaction with a host cell. During HIV replication, more than 250 human genes are involved in viral replication. And those 15 viral proteins can interact with more than 400 human proteins. Those are human factors to support HIV infection, but in our body, we have very powerful immune cells to fight and control viral infection, including virus, like HIV. For example, this is a special group of white blood cells, our strong immune cell to recognize the invading pathogen and elicit our immune responses. When HIV infects those dendritic cells, the virus can get into the cell, but the viral replication is stopped inside. This is a very fascinating phenotype for our scientists. My lab has been studying HIV interaction with the dendritic cell for over 12 years. Many scientists, including us, want to understand this mysterious anti-HIV function inside of a cell. When we use this special power to control HIV infection, or maybe develop a very effective treatment. But it turned out the puzzle is not that easy to solve. Only 6 years ago, scientists discovered how does HIV get inhibited in this special immune cell. When the HIV particle gets into this immune cell, the viral RNA needs to be converted into DNA. But we have a superpower soldier in our cell, Captain America. He can fight and control HIV inside of this immune cell, and the viral DNA replication is stopped. So it turned out this super soldier is a human protein called SAMHD1, named after the protein functional domain. So what is SAMHD1? How does it work? To answer those two questions, we need to understand how does HIV replicate in an immune cell. In our cell, we have those DNA building blocks called dNTP. Those are used by our cell to synthesize, to make our DNA, which is very important for DNA structure and function. You can imagine those are like bricks to build a nice house. However, HIV steals dNTP from our cell to make its own viral DNA during the viral replication. What does SAMHD1 do? SAMHD1 turned out to be a very unique enzyme. The enzyme can break down dNTP in our cell by removing this triphosphate group from dNTP. You can imagine it is powerful molecular scissors to seek the balance of dNTP in our cell, which is very important to keep our cellular function. However, when HIV infects the immune cell, during the viral replication, it steals our DNA building block, dNTP, to synthesize viral DNA. SAMHD1, as a super soldier, can block the DNA replication. You can imagine SAMHD1 acts as a pair of pliers to cut the power to HIV, which is the dNTP, the DNA building blocks. In our laboratory, we can review the function and the mechanism of SAMHD1. As you can see here, when we infect the dendritic cell from our blood, the viral infection is well controlled by SAMHD1 in this cell type. However, if we remove SAMHD1, the viral infection can jump over 40-fold. Similarly, you can see our dNTP level in those cells are very low because SAMHD1 breaks down certain level and which is even lower than HIV-required DNA building blocks. However, if we remove SAMHD1, then the dNTP level jumps more than 20-fold. Now we know SAMHD1 can control HIV infection by decreasing dNTP concentration in our cell. But the question is, can we translate this into a therapy? Can we develop a new drug based on this very powerful enzyme in our cell? So the idea is, can we starve HIV in those immune cells, HIV targeted cell, and fight or eradicate HIV infection? Interestingly, several groups, including my lab, we discovered a molecular switch. We can turn on SAMHD1 anti-HIV function better. And also we knew how to increase this very powerful enzyme in our cell. We hope one day, with many different novel approaches and our research on SAMHD1, we can achieve a cure for AIDS. I hope I will see this in the near future. If I'm not retired, I will continue SAMHD1 research because this enzyme is very critical for many functions in our cell. For example, it balances dNTP pool in the cell. It also restricts not only HIV, but also many human and animal retrovirus and other DNA viruses, like human hepatitis B viruses. Finally, SAMHD1's major function is to maintain genome stability. Unstable genome can lead to very severe innate immune disease, autoimmune disease, or cancers. We are now collaborating with physician scientists and cancer researchers. We try to contribute our work to better treatment of those very serious diseases. Thank you.